Thanks everybody for joining us today. These are our favorite calls of the week, especially when we get to have special guests. We got to hear from Daniel a few weeks ago, and now we get to hear from the fabulous Dr. Knox. I had a great conversation with them the other day, kind of diving in more to the opportunities and the way that we framed this conversation is just to go deeper in the data and the history of this virus so that you guys have an opportunity to ask all your medical, scientific, and data questions. Use the chat or pop in. I don't think that she would mind if you interrupt, the flows.
Oh, absolutely not. I think it's always
Yeah,
better to get a burning question out of the way,
right.
before you proceed, so things are clear.
Yes, that
Mhm.
sounds good. So we'll let you take it away. You have share screen rights so that you can share.
Well, let's see. Oh, recording, view, share screen, share.
Yep.
Okay. Screen. I'm gonna do screen two, right? Okay. Okay. Can everybody see?
Not yet.
No.
Oh, screen two. Oh, share.
Perfect.
Okay, good.
Yes, we can
Good.
see.
Okay. So this is me with a different haircut a few years ago, but it is me. And so I am Connie Knox. I am the founder of COPA Health and COPA Laboratories. And my background is my training is laboratory medicine, but it is really pathogenesis. And so pathogenesis, especially as it takes us into infectious diseases of humans and of animals. We have done this little HHV-6 virus has taken us on a long journey starting in 1987 when we isolated it from a patient presenting with pneumonitis and severe neutropenias. And we felt at the time he was HIV/AIDS, and diagnostics for HIV was challenging back in the late 80s. And as it turns out, the virus we isolated was HHV-6, and we found it in lung biopsy of this. That kind of launched us, and frankly, our bone marrow transplant program into thinking about marrow suppression syndromes in the BMT. We weren't stem cell transplant at the time, so we were truly BMT. So I'm gonna spend a few minutes on that and why we think it's relevant for the infertility space. So the outline, we're just gonna spend a short period of time on the basic biology of HHV-6 and how it may contribute to reduced fertility in women and men, the laboratory tools that are available for diagnosing HHV-6. We'll talk briefly about our collaborations and our conference presentations that we've had with some of our REIs, and then what kind of antiviral intervention options we have for HHV-6, and then Alice's favorite, the ICI, or inherited chromosomal integration, that has some association with preeclampsia. So human herpes viruses, we've known about, they're more than 1 million years old. They were first discovered or documented by Hippocrates 400 before Christian era. But HHV-6 was more recently discovered at the National Cancer Institute coming out of a lymphoma AIDS patient. And that's less than 40 years ago. And we got involved. This patient I described to you, we found in 1987 because we thought he was HIV. But most of our work in the late 80s and early 90s were really related to HHV-6 in the bone marrow transplantation space. Background, HHV-6, like the other herpes viruses, establishes a primary infection, and then the infection is lifelong in the host. We suffer with periodic reactivations of the viruses. In immunocompetent patients, those are more annoyances than severe, but in immunocompromised, HIV/AIDS, bone marrow, solid organ transplant, autoinflammatory illnesses, they can really be quite serious. And so we'll talk a minute about the HHV-6 in the bone marrow transplants and why we feel that HHV-6 may be also important in the uterine microenvironment and perhaps contributing to interference with the early stages of embryo implantation. So these are just a few of our publications from the early 90s at the medical college. We've got to the right are the clinical reports and what was consistent in being able to isolate virus from blood or bone marrow in the bone marrow transplant patients was consistently in the clinical context of failure to engraft or marrow suppression, incomplete reconstitution of the various bone marrow lineages. We have also several publications with our in vitro studies. And again, what we saw with HHV-6 was when you infect bone marrow stem cells, we would see a failure of outgrowth of the different lineages, and also, interestingly, a failure to respond to growth factors. That was also a clinical observation where patients who reactivated HHV-6 at 2 weeks after transplant, that was the most common presentation. Their marrow, we had various lineages. The granulocytes were a big one, and platelets were a big one that would not reconstitute. And sometimes the graft was a total failure. So this is so how is an embryo like a bone marrow transplant? Because of our transplant experience, my feeling when the reports of HHV-6 in the endometrium and the association with, again, pregnancy failures or in patients who had infertility for unknown reasons, it was like a light bulb. I mean, it just seemed, boy, we've seen this before in another context and not in an animal model. We saw it in a human model. And we've often kind of thought of our transplant, our iatrogenically suppressed patients as experimental models. What can happen what would these agents do? What do viruses do when the immune system is not there to prevent the virus from really going unedited? So my thoughts about this, why are these similar? Well, because both bone marrow transplant, normally, if it's allogeneic and an embryo, they have genetic material that's foreign to the mother or the recipient. It requires a state of immunosuppression or immunologic tolerance to avoid rejection. It requires the stem cell or the stem cells to be able to migrate to a specific location in the body, and it requires a receptive microenvironment in order to get established, to differentiate, and to proliferate. So that is my personal sense of why we feel that there's a personality to HHV-6 that lends itself to this kind of activity, which is its blockage of hematopoietic, or in this case, the embryonic stem cells to get established. Most people are pretty familiar with the herpes simplex viruses. These 3 families of herpes viruses, the alpha, this is chickenpox, Down here, we have our oncogenic viruses, EBV and Kaposi sarcoma virus. And right here in the middle are a group of viruses, especially CMV and also HHV-6A and B, that cause trouble in immunocompromised patients. Genetically, we are called beta herpes viruses. That includes HHV-7, which we haven't really figured out where HHV-7 lends its trouble, but HHV-6 has been a virus of special interest to us, again, going back to 1987. Now, there are we used to call them variants. They are now determined to be really genetically different species of HHV-6. They're about 90% genomically identical, but they have really different biologies. HHV-6B is known as the roseola virus, where most children by the age of 2 have become infected. And to a great degree, that is because the virus replicates in the oral pharynx and is easily transmitted to children through just affection from their caregivers. So we know a lot about HHV-6B because we can easily isolate it from saliva, from blood. It's been really simple to study. In contrast, HHV-6A, we don't really know that much. We don't find it in blood, very rarely in a very severely immunocompromised patients. AIDS patients we can find HHV-6 in the D of those. It's not found in saliva. We don't know what the prevalence in the population is, and transmission remains uncertain. More recently, and this will move now into what's been kind of published and discovered in unexplained infertility. Infertility, early miscarriage, and fetal death. There have been reports that HHV-6 can be found in association with those clinical presentations. I suppose the landmark publication for HHV-6 was in PLOS ONE 2016 by Marcy et al. It's a group of Italian investigators, extremely talented group. These labs were some of the very first back in the late 80s and 90s that were studying HHV-6 primarily in the context of HIV/AIDS. Because we were all young bucks back then, sharing data, the old investigators, which I am included, we had a very close working and collaborative network. And this group of Marcy et al. Are still part of people that we talk to and people that we can go to. Hey, what are you doing? What have you seen? And, what do you have funding for? But theirs was the very first publication looking at unexplained infertility in women. And their results were that 43% of the infertile women in their cohort had HHV-6 detected in endometrial biopsies. 0% of fertile women, which were defined as women with 1 or more children. And you can see there were different perturbations that they also noted. Hormonal levels that were changed. The endometrial NK cells were lower in number. The distribution of the cells was different. And then also cytokine perturbations were noted in these samples. And so their summary after this study, they proposed that HHV-6A appeared to alter the microenvironment in the uterine endometrium. causing a cascade of changes in this microenvironment with respect to cytokines, growth factors, hormones, and cellular composition. But what's interesting is there were no obvious clinical signs of infection in these patients, and virus HHV-6A was not detected in the blood. They had a follow-up paper just about a year later, looking again at endometrial biopsies and uterine flushings, and analyzed HHV-6A by real-time PCR and immunofluorescence, and they were checking out again the endometrial NK receptor. So they're now kind of delving down into more of the cellular interactions, right, to see if they could determine and identify disruptions. The most interesting thing to me that came out of this paper was the fact that they again replicated, they had 40% of the infertile women demonstrated HHV-6A only in the endometrial epithelial cells. And you can see here, HHV-6A was found in 8 of the 20 infertile women. The virus was not found in endometrial stroma. It was not found in endometrial leukocytes, and virus was not found in peripheral blood mononuclear cells. By contrast, HHV-6B was not found in either infertile or fertile women endometrial samples, and not in stroma, not in leukocytes. But because we know HHV-6B can like CD4 mature CD4 cells, they did find it equally in fertile and infertile women. So this last sort of major, I think, or important publication by Dr. Carolyn Coulomb, she analyzed retrospectively, she had frozen biopsies from recurrent implantation failure individuals from her practice. These had 4 or greater failed embryo transfers, 10 fertile women, and then she analyzed or tested for HHV-6 by an mRNA assay and by sort of their specialty endometrial immune profile. And again, consistently, over a third of women with recurrent implantation failure were positive for HHV-6 nucleic acid, while 0 of the fertile women were. There is a little bit of data for HHV-6 being present in male semen samples. And this was a Danish study of about 200 sperm donors, and they looked for all of the herpes viruses, and you can see that they had 55 of the donors, or more than a quarter of them, had detectable herpes virus DNA in the semen sample. And what is interesting, again, for us, seeing as we're trying to figure out how does HHV-6A, where does it come from and how does it move around? It was the dominant herpes virus found in these semen samples at about 13% of this 27%. They took it a little further and did a little bit of in vitro investigation, mixing sperm cells with virus, and they found that the HHV-6, in this case it was B, but HHV-6B was able to adhere to the acrosome of the sperm head. And this then suggested to these investigators that HHV-6 has an opportunity to be transported into the uterus via binding to the acrosome of the sperm head. And this kind of now makes sense that we have we have the coupling of two different reproductive systems and the sharing of biological fluids, that this is a reasonable way for a herpes virus to be transferred within the population. What do we do for diagnostic testing? Well, we at COPA, we've been doing immunohistochemistry testing for HHV-6 from, oh goodness, from, again, probably the late 80s. Most of the populations we have tested are biopsies from, again, immunocompromised patients, be it AIDS patients, solid organ transplant, bone marrow transplant, a lot of children's samples, acute liver failures, etc. So we have a very well defined assay system. And so when in 2017, I was contacted by two REIs who had seen the Marcy paper. They had seen the Italian paper, and they were interested in looking at some of their patients and having some of their patients tested for HHV-6 in endometrial biopsies. At the time, I was not even aware of the paper because our laboratory had done some refocusing on tick-borne disease. I again was very interested, but kind of a little bit surprised to learn that my Italian collaborators had done, such a fine study. So that's you. Okay. So this is the assay that we use. Our antibody is directed at a late structural protein of the virus, which means the virus is in replication mode. So our test tests for active infection, which is important in the transplant setting because the presence of DNA for a herpes virus does not necessarily mean that the virus is active, because we know it can sit in latency. So if you're going to make a decision about treating transplant patients with some of the bad drugs that we have to use for HHV-6 in those patients, you really want you want to have confidence that you're actually treating an active infection. So I'm just going, okay. So one of the studies that we did that was presented at, I think this was SRI, or maybe it was ACOG. I do have for the 5 studies I'm going to describe in briefly, we do have the posters that were generated, and I'd be happy to send out the files for that. But we were interested in looking at different kinds of infertility patients, and certainly endometriosis is a factor in infertility. We didn't know if HHV-6 would be found in the endometriosis patients. So in a collaboration with Mike Duty, who has a lot of endometriosis patients in his practice, we looked at 17 patients, and we had the endometrial sample plus the peritoneal implant. And what we found that 3 of the 17 individuals were positive for HHV-6, and if they were positive both in the implant and in the endometrium. We subsequently got another 8 samples from Mike and had another additional set of cases of patients with history of endometriosis, and that we didn't see as much in those cases. So our data for HHV-6 and endometriosis is about 4 cases in 38 women, or about 9%. So that's much lower than we're seeing with just the infertility with the unexplained infertility, and certainly much lower than what we see with in the patients with recurrent implantation failure. But it is found in those patients. Its contribution to their infertility, I think we just don't know. There was another set of studies that we did with Zitao Liu out of New Hope Fertility, and this was retrospective analysis of 37 women evaluated for infertility and for potentially for IVF. I wish I had a pointer. I know I can do it, but right now, if we look,
your mouse around if you want, so that I think if you move your mouse.
Can you see it? Oh,
I'm not sure.
You can see it.
Okay, yeah, okay. This study, it's interesting because, again, even though it's low numbers, out of 37 patients, we saw 10 positive in the endometrial biopsy for HHV-6. But again, in the patients with frozen embryos and failed transfers, we were at 50% in the HHV-6 positives, and in only 6 or 22% of the HHV-6 negatives. And with euploid frozen embryos, again, it was 40% versus 18. This is not, the numbers are just not statistical. But if they're a little bit more, I mean, we're at 0.1, so I think it's a numbers thing, and it's just consistent with what we see. But what Zitao did see was that the presence of HHV-6 was strongly associated with endometrial polyp and adhesions. So of the 10, 9 of the 10 HHV-6 positive samples, and then only 9 of the 27. So this was something we had not been sensitive to before. So again, the correlation, is it causation? Of course, we don't know, but again, there seems to be a difference when HHV-6 is present in the endometrium. This ACOG, this is sad because we actually didn't get to go to the meeting because it was in April of 2020. So COVID just kind of threw us under the bus. And Katie Duty, I think she was in her last year of OB-GYN residency, and she was very excited to present it, but we didn't get to. But Katie Duty was taking some of the data from her parents' practice. And again, if we look just at, in these 200, there were 200 women, 75 met the criteria for repeat implantation failure. We look at that and we're seeing 51% of those with repeat implantation failure were positive for HHV-6. In the patients with unexplained or recurrent pregnancy loss, we still see a sizable amount, but I suspect that these patients are enriched out of this group. Maybe they're not the same, and maybe those with more knowledge can say, no, they're really different populations, but the more we look closely at women who can't keep an embryo, the more we see the frequency of HHV-6. This was a study we did with Bert Scotcha at University of Illinois, Chicago. This was kind of an interesting study because these were women with their first encounter into the UIC program. So I think this was even these biopsies, I think, were even from mock cycles, and then the transfers would be done the next time. And I think that's not uncommon. But And Bert's interest was more focused on endometritis. So his thought was, in these patients with endometritis, and sometimes they don't find a pathogen, could HHV-6 be driving the inflammatory response? And as it turns out, we did not see an association between HHV-6 and the presence of the endometritis marker CD138. But what we did see was endometrial biopsies. We had 35. 6 of the 35 were positive and 29 were negative. And again, these were really this was a heterogeneous group of women with fertility problems. With the HHV-6 positives, we had data that 5 embryos were transferred, but only one patient was able to keep the embryo. And the live birth rate was 20%. That was much lower than if the patient did not have HHV-6. We saw in 25 embryo transfers, 15 of the 25 became pregnant for a success rate of 60%. Again, numbers are low, but the presence of HHV-6, this is untreated. So this was kind of retrospective. They didn't know, they had not looked at HHV-6 before that. So the presence of HHV-6 looks to be a predictor of bad outcome. And if we kind of summarize the prospective study, and which was the UIC study versus the duty study with recurrent implantation failure, we see that only 17% of prospectively collected samples prospectively collected samples from women who had undergone embryo transfer, only 17% of those patients got pregnant versus 50. Wait, am I reading this right? Be prospectively collected. Patients okay, this is again for enrichment of our failure group. You can see more generalized population is 17% HHV-6 positive, and those who have that repeat failure of the embryos to be able to get established is again associated with HHV-6 at a very significant level. This is kind of the last study that I wanted to just share with you, and it really is a setup for antiviral treatment. This was a study we did with Kevin Duty, and he had 51 patients, again, with unexplained repeat implantation failure, and he was doing serial biopsies. So he had 26 positives, and he treated these women with varying levels. First it was acyclovir, then we talked and he went to valacyclovir, and then he was increasing his dose because he was trying to get the biopsy to turn negative. He was trying to erase the red, which is our colorimetric reaction for our viral antigen. And here is a picture of that. This was a patient who cleared after the first set of treatment. It's a little harder to see, but the red antigen connotes HHV-6 late glycoprotein. Here it's cleared up, but in some of these patients, you could see that there was over time a progression to antigen reduction. And here is kind of how we landed on the that valacyclovir at higher doses was looked to be kind of in the ballpark of what we needed to do. Now, acyclovir, valacyclovir has got a great safety profile in pregnancy. A lot of data that showed basically no significant negative impact in 1,800 infants versus 20,000 infants without exposure to acyclovir. One interesting piece of data from our old transplant days was that the use of high-dose acyclovir in right before bone marrow transplantation demonstrated a significant reduction in HHV-6 reactivation. And I think that's what the goal is here, right? Acyclovir is not real strong drug against HHV-6, but in pregnancy, we don't really have another option. We can't use foscarnet, cidofovir. Those are all teratogenic. Those would not be available in this clinical setting. So the goal would be to get antiviral on board to prevent the virus or suppress the virus and keep it from reactivating in this patient population. These are valaciclovir, herpes simplex, really, really sensitive herpes simplex. And for HHV-6A, it's not bad. So it is within kind of the therapeutic window. For HHV-6B, it's a little bit different. It's about 3 times. The IC50 is about 3 times. So I think we're actually kind of lucky if we're thinking that only HHV-6 is the problem in these infertility patients. Pregnant women with recurrent genital herpes simplex, again, I think it's pretty commonly used. I guess the current recommendation is Valtrex 500 mg twice a day. I have spoken with, this was early, there were 2 REIs who were using early on in their bid to suppress HHV-6, they were using acyclovir. We wound up kind of increasing the amount of the acyclovir availability, and that really with Valtrex, the val ester of acyclovir gives you a much better bioavailability, and the dosing at twice a day is so much better than with acyclovir. It's 4 times a day. It's very hard for patients to be compliant taking pills 4 times a day. And so that's, you know, this looks to be probably where we want to go. And multiple REI practices are now pre-treating their HHV-6 documented patients for about a month before an embryo transfer. And in discussions with them, it's like, well, we've had these conversations. How long should I keep my patients on these? And my analogy is the bone marrow transplants. If you can get the graft, and the embryo to me is like a graft. If you can get the graft to stick, after that, in the bone marrow transplant patients, they had pretty smooth sailing. They did really well. So there was just this window of vulnerability when those stem cells are trying to, you know, kind of adhere to their matrix and get going. If the virus reactivates, it interferes with what the stem cells need to do. And so, as I mentioned, we were really cooking along and doing some very, I thought, important work with the REI IVF clinics in this field. And we had 3 presentations at 2 different meetings scheduled for March and April of 2020 that were published because these meetings were canceled. The symposium for what is it, reproductive investigation, I think, that I was supposed to, my flight was on Friday, and they canceled the conference on Tuesday. It was the middle of March, And then, of course, the ACOG. And that was Zitao, and Zitao being Chinese. I mean, all of this COVID stuff coming from China, and he was very nervous about it. And the meeting was supposed to be in Vancouver, and that's where there was kind of the local outbreaks of SARS-CoV-2 then. And I was trying to convince him that it wasn't, oh, it's not that bad. It's a respiratory virus, we're healthy. And then they canceled it, and we didn't really have to make the decision. So I would have gone Zitao, I'm not sure, but we didn't really have that choice. So, but the data that we do have for outcomes, because ultimately it really comes down to, if you have a test and you identify a factor and you do an intervention, in this case, it would be antiviral treatment, what changes? And we want it to change for the good, right? So there is a lot of data actually out there in these IVF practices. I think there's probably, I would guess, 150-ish patients who were treated, and there is outcome. But that was like, multiple years ago, and it's been difficult to convince the IVF clinics to go back and pull the outcome data. But what we do know is, if you are HHV-6 negative patients or negative biopsies with, of course, no treatment, the pregnancy, the implantation rate was 58%, which I think is kind of the norm-ish. In individuals who we identified as HHV-6 positives, but those were retrospective, so we didn't have the data at the time of what their biopsies were showing. There was only 1 of 6 that we're aware of that became pregnant, and that's 17%. So if you have HHV-6 and it's not treated, the success rate of implantation in the IVF setting is not good. In those patients who were 6 positive and were treated with antivirals and at a level that was effective, we kind of return to the baseline success rate that is seen in these IVF clinics. So, and again, there is more data out there, but this is positive. This is what I've been able to convince the REIs to share. This is one last thing that's now, we got to step back, a little bit divergent, and it is the concept that human herpes virus 6 integrates full length, its full viral genome into human chromosomes. There is not another herpes virus that is known to do this. And this was actually discovered just like 25 years ago. And so this inherited chromosomal integration in the fertility space has been associated with preeclampsia. And the chromosomal integration into the germ cells is believed to be, the data shows it's in about 1% of the human population. Some areas of the world have a higher integration rate, like the Mediterranean and in Africa, where we think HHV-6A really kind of started a long time ago. But both A and B can integrate and do integrate. We do specific testing to confirm it. We do hair follicle analysis to preclude any kind of contamination from blood or other. So this could be something I think that should be discussed, because it may be something that clinicians should be aware of. And in our menstrual fluid tests that we currently are just starting to, try and educate people about, the menstrual fluid molecular test for HHV6 will identify to a very strong degree the chromosomally integrated patients. It's a feature of how the test is designed. So we can identify those with strong confidence, and then a confirmatory, again, would be a hair follicle or nail clipping. We prefer the hair follicle because sometimes in families who have chromosomal integration, if we get fingernails, the skin cells from just people having personal contact, those can be contaminated. So we prefer the hair follicle. And it's a curious thing. We don't understand why the virus, but it's the structure of the viral genome of HHV6 genome allows for relatively efficient integration into open or replicating DNA. And it looks like it could have some involvement in pregnancy. So this was a beautiful fall day. It wasn't winter. I didn't take the picture in like March, for example. But that's us, and I appreciate the time today. Thank you. Thank you.
What questions does everybody have? We're going to get the
Connie,
desk here, right?
Connie, thank you for your presentation. I know Kevin Duty very well, and I'm curious if, since I know that what you were doing with him was several years ago, what is he doing now, and is he doing the surveillance for HHV6 now?
Following COVID, our lab actually, we got into the COVID business very quickly. We had a test on board by the middle of the second, third week of April. Kevin's group, I'm sure, like most of the IVF clinics, just were trying to figure out how to continue to do their practice. So everybody got distracted in a different fashion. I had contact with Kevin the next year, and we talked about gathering up all the data and getting the data, together in some sort of manuscript fashion. But I have not been able to, I've not had conversations with him since then. So as far as I know, I don't know if he's somehow doing other kind of testing. The only lab that was doing HHV6 testing, and they're not currently doing, was the Rosalind Franklin group. And they were doing, that was the lab that Dr. Carolyn Coulomb was working with. And they have the test offline at the moment. So I'm just not familiar with where Kevin is. I've tried to reach out, but I just haven't received any feedback from him.
Now, I know Carolyn Coulomb passed away, but do you have any idea why her lab at Rosalind Franklin is no longer offering the test?
I do not. I know they contacted us about within the years, within the year, and they were they wanted to buy viral control material. We don't sell controls. I mean, we're not a distributor, and they were commercial. I suggested to them that they contact the HHV6 Foundation, because the HHV6 Foundation has cultured material. I think they have nucleic acid. I don't know exactly what they needed, but I suggested she contact Kristen Loomis, because they do have resources. It may be that the field just moved on, because that happens. The field just moved on to other more immediate, sort of things. Because this was grassroots, right?
Mhm.
There wasn't a budget for it. There wasn't specifically, money for it. So the REIs, the IVF folks, it was their time, their commitment of staff. I think that the COVID break, we just lost momentum on that.
Can I ask one more question? Do you know how I did the EIP, the endometrial
Immune profile?
immune profile with Carolyn Coulomb, probably on about 30 patients over a span of maybe 3 or 4 years. And she had a protocol
Yeah.
of whether the response was too high or too low, there was a
Mhm.
certain treatment. Do you know how that test was or how that differed from what you're measuring with HHV-6?
Yeah, they had their test, as I recall, it's like kind of like the ERA test, right? There's just it's an expression analysis. So they're checking for mRNAs in the sample and hoping to identify or hope the goal was they would establish parameters for what would be a good heat map on an individual basis. So when to do the transfer, right? It was all about trying to optimize the embryo transfer. And that is a really tricky thing. And the ERA is, I don't know, they've got hundreds or 1,000 genes. Carolyn's again was an expression analysis, mRNAs, to try and identify optimizing again what I understood to be when is the best time to do the embryo transfer on a specific patient by patient basis. And it's just really hard because there's so many things that can perhaps change the expression. And even ERA, that was a big program. There were big studies and they were conflicting. And the IVF folks often had their own protocols that really worked pretty well with them. So again, trying to by messenger RNA, try to figure out, optimal embryo transfers. It's just, I think it was just hard. And maybe the data just was never really clear cut. There was always controversy with those systems.
Okay.
So ours is simple because We're just saying we think that there's a virus. We can detect the virus. We know if the virus is there or not. And we think, by analogy, that the virus may well be interfering with those early attachments, whether it's because it abrogates the effect of, the natural cytokines like GM-CSF, which the endometrial cells, the epithelial cells, produce most of it in the uterine cavity, or by other things, adhesion molecules. It messes with that, or this is this virus. It's pretty simple. We're just saying when the virus is present, this is kind of the outcomes. These are the failures that we're seeing, and it looks like pre-treatment prophylaxis with Valtrex helps to quiet down the virus and allow for better blastocyst attachment. So we don't have a complex algorithm. It's really simple. It's just really simple. And that's what we found in the BMTs when it came to HHV-6.
In somebody who has true RPL and not like recurrent chemical pregnancy loss, where they get a positive at-home pregnancy test and then not a confirmed heartbeat. Is there any logic to keeping them on the protocol longer through the pregnancy, like until, second trimester or something?
We have no data to know if that would be helpful or not. I mean, I think the good thing with the acyclovir is its safety profile. From
But would you
current,
start
the
with
current,
Valtrex and then switch to acyclovir?
well, Valtrex is acyclovir. It's just kind of packaged a little bit differently
Oh, I see.
so that it's got a higher bioavailability. It's the same drug. Valtrex is
Got
a
it.
prodrug of acyclovir.
Gotcha.
So it's the
Okay.
same. It's the same thing. And the patients are remanded to their OB-GYNs at after heartbeat at 3 months, 2 months, or
Usually
whatever
week 9.
this.
I mean, usually in the IVF setting, it's still week 9, right, Lori?
Okay, okay. And so the REIs that we still work with, they've kind of landed on that 6-8 week heartbeat thing for the
That makes sense to
Valtrex.
me for like implantation failure patients and people like me who end up, getting pregnant and losing very early, by like week
Mhm.
6, 7.
Yeah.
But it's a curious thing, and maybe that's a study that we should be orchestrating, with some IVF partners ourselves too, kind of in coordination, where somebody who has made it farther than that beyond week 12, if they continuously, if they have a number of pregnancy losses past week 12, where it's because isn't getting through first trimester then make it recurrent pregnancy loss, like the chemical pregnancy losses don't really count in clinically, correct? I'm asking my docs to confirm my understanding. I
Yes.
feel like, yeah, okay. So I'm just wondering, yeah, if we should look at doing that study with ourselves. I mean, we need to be good stewards of the data tracking anyway, but I'm wondering, even with all the IVF docs that have helped fund Ovum through MedAnswers, if they would all be willing to do a study with us. Wow.
Yeah.
Uh-huh. Right. Is a woman, and forgive me, docs, for asking something that might sound like a dumb question for you all. Are we immunocompromised just by virtue of the pregnancy for the entire pregnancy, or is that just like the early stage? Is it entirely like the whole entire time?
Well, you see an increase in viral reactivation. So the immune system is dampened to some degree. I kind of think of it, I know there's the reactivations, especially third trimester. I'm not sure, maybe, I don't know, is Sue still on? Nope. Sue
She's
must
on.
have-
Yeah,
Oh,
yeah, I'm here.
yeah, so what's the data with like herpes virus reactivations during pregnancy? When does it peak? I mean, progesterone gooses the virus, so we know it kind of likes that change.
What does that mean? Is that a scientific term, that it gooses the virus?
Oh, yes, it
can be.
tickles it so that it becomes more active.
Oh, progesterone does, because
Right.
we do a lot of
Yes.
progesterone supplementation until week 9.
Yeah.
So
So
maybe- yeah.
yeah, in pregnancy, when you guys see these, when is the concern about viral reactivation in pregnancies or in some of the pregnant patient or babies that you guys would have seen at Children's?
Well, if we're looking at herpes simplex viruses,
Yeah,
then the concern is obviously there's some concern early on and then again at the time of delivery.
yeah,
Because you're expecting a vaginal delivery and they're worried about transmission
yeah.
to the
Right,
fetus
right,
unit,
right.
Okay.
Right.
Understood on that. And so, but have you seen that that there's high reactivity during third trimester and we don't really know why? Or is it just, yeah, okay.
Yeah. Yeah.
Yeah. We
And
also
we
know
don't know
that,
if there's any transmission like through the placenta for the genetic form of HHV-6. We
there's,
don't have any idea how that
yeah,
happens.
I've seen 1 to 2%, 1 to 2% transplacental infection with HHV-6. Sometimes though, you got to look and see if that's old data, because it could have been chromosomal integration before we understood chromosomal integration.
Right.
So I think there's a lot that's just kind of, not been nailed down. We just don't have confirmation. I think we don't
That
know.
chromosomal integration stuff, that's creepy. Am I the
It
only one who
is-
thinks that way about it?
It is weird. It is
Yeah.
weird. And some people believe that the virus can pop out of the gene. It's like jumping genes, right? They pop
Wow.
out, they circularize, and they can make RNA and protein and stuff. I'm not sure I'm convinced on that data, but what is clear, it is likely if the herpes virus genome, if it's in frame, right? There's no reason why messenger RNAs and proteins, why there couldn't be transcription and translation of some of those genes. So I think that's kind of something most people are comfortable with, because you think about it, it's another gene, and it really depends on how it orients in the human genome. And then what do those genes individually, like EBNA1 for EBV, EBNA1's about all you need to transform a B cell. You don't need replicating virus, you just need the gene product. So does HHV-6 have gene products that can do things even in the absence of full viral replication? Could well be, especially for when the virus is integrated.
Yeah,
What
can I,
do we tell a patient if they have that? What do you tell them? Sorry, Lori.
go ahead.
What do you tell a patient if it's integrated? Do you still treat them for the activated form that we found in their menstrual blood?
Well, most people who have chromosomal integration, most people probably, well, for one, don't know it, would never get tested for it. We've kind of argued for it for pre-screening transplant and recipients, because I will say that chromosomal integrated, if you're a transplant patient and you get an organ, you get a bone marrow transplant or you get a liver from a patient with chromosomal integration, when we're doing surveillance for HHV-6, there's so much DNA. I mean, it's like, whoa, is this patient having a roaring infection? It confounds laboratory testing, and it's a real pain.
Wow.
And it would be simple to check the donor and the recipient to see if somebody's integrated, because the hardest thing is when you have a hybrid transplant, right? You have a high level, but it's not high enough to be an integrated patient. Well, is it active or is it not? And I will have to say, we have gotten multiple biopsies from clinicians, especially children's hospitals and transplant, trying to figure out, does the kid have an infection or did he get an integrated graft? And it's easily so. I know HHV-6 Foundation has really, lobbied for that, but it's not, again, it's just getting things on protocol. If it's not protocol, it doesn't get done. So there just has to be buy-in from the different agencies that, take care of these patients.
Lori, what were you going to ask?
I was gonna just make a comment that I see some strong similarities in terms of how reproductive endocrinologists view this particular test to be very similar to their bias against checking for natural killer cells.
Oh,
That's been very controversial for decades.
Yeah.
And I think, for example, out of 11 physicians at my old practice, Fertility Centers of Illinois, I was the only physician working with any of the reproductive immunologists, including Carolyn Coulomb, because I felt that she was very legitimate compared to some of the others who were looked at as charlatans, quite frankly, in this particular field of reproductive immunology. So there's never been a universal acceptance
yeah.
of immune factors as it relates to recurrent pregnancy loss. I think in large part because some of the early pioneers in that area perhaps were either charlatans or they overstated their importance. In other words, I know for a fact that one of the players in the Chicago area, immunologist back in the 80s and 90s, felt that virtually every miscarriage was related to immune factors. And we know that's not the case, but it put a bad taste in people's mouths. And so I think you're going to find a natural reluctance against something like this. Although I admit I did check natural killer cells, and similar to the HHV-6, you have to diagnose it with a uterine biopsy, not in the blood. In the blood is going to be very unreliable.
Right.
But I think it's going to be difficult for people to accept this. Now, perhaps we can be in the forefront and use this to help our patients, but you're going to see a lot of skepticism from reproductive endocrinologists.
Until the numbers get bigger, I'm sure.
Yes.
I think it's, I would guess, or I would think that when there's enough outcome data to be more compelling, and I think that's numbers, because my question would be, checking the ENKs, if there's a perturbation in that cell population, is there something you can do about it to kind of normalize? I mean, is there it could be there's perturbations because there's a virus floating around, or perhaps a bacteria. I would bet on a virus, but some things are
That would be interesting
of
study,
academic interest and important to know, because eventually we may find an intervention, but that's been, the NK cell function in chronic inflammatory diseases or chronic fatigue syndrome. It's noted, but it's really hard to come up with a therapy that is consistently, I guess, re-regulates them.
Right.
It's not a bad
Well,
thing
I think
to know about, but what can you do?
Well, an advantage, I think, of this particular way to detect the HHV-6 is that it can be done without the biopsy, and so you don't have, the patient doesn't have to go through the biopsy, and also then the treatment is, not it's not invasive. There's not a lot of side effects. It's pretty mild. So it's an easy thing to actually be able to implement and get some data on really outcomes, and how well does just being able to find it in the menstrual fluid and have your patients not have to go through the biopsy.
Yeah.
Granted, I know that many times you're going to go that route anyway because of all the other things you want to look at.
Not us, we're telehealth too. We won't be doing biopsies online just yet.
Okay. So the fertility centers will be doing the biopsies on it, but maybe they want their patients to go that route so they can look at a bunch of other things.
Well, exactly. If they're already
Besides just this, which I think is an easy, non-invasive way to be able to look at it and be able to look at outcomes.
Well, and if you really want to get, to if the patient's on treatment and is actively trying to conceive, and if they have another period and they've been on treatment, you can collect for another evaluation to see if because our assay is quantitative, so we can see if there's a drop in load, right? If the patient were positive in the first biopsy. So we have a way perhaps to better predict outcome. That one slide set I showed you with Kevin, I mean, there was, doing multiple biopsies to try and check for diminution in antigen is not preferred unless you're doing biopsies for another reason. But to collect another menstrual fluid just to see if, I was positive at this level and now I didn't get pregnant, but it looks like the level's dropping, or just to check. I mean, it's an easy way to confirm that the antiviral is having some effect.
Mhm.
Well, my ultimate dream, as I think my team knows, is that one menstrual sample can run a carrier screening, an HHV-6, and the endometriosis non-invasive test as well. That's my big dream. So I've introduced Connie to Reedy at Next Gen Jane on that side. Reedy was already on board to use her tampon, tampon collected sample for carrier screening. We just didn't have a lab. So hopefully Connie can be the lab and we can accomplish actually all the testing we need. I see the same thing being possible on the male side with one sperm sample. If we can run carrier screening off of one sample, plus the molecular sperm test, plus an HHV-6, then we have such advanced comprehensive analytics for our patient population, and they don't have to go do a whole bunch of different types of samples. I mean, those 3 tests alone would be menstrual cup, menstrual tampon collection, saliva, semen, and we still have to send them for, blood work. So it's just, it's just a patient hassle. So that's my big dream that we hope to make come true.
You didn't mention hair samples.
I did not. You're right. We have a hormone hair test.
You
Since
do?
you need, so since you need the follicle, our hair test didn't need the follicle, but I'm going to need, I forgot to mention that to you, I'm going to need your lab to run our hair test too. So since you do need the follicle, it's fine. You don't need the rest of we don't need the follicle. We just need the rest of the hair, but it's the only non-invasive way to measure AMH. Now, if we're collecting menstrual blood anyway, we could just run, all the same labs off of the menstrual blood anyway. But yeah, AMH, we tried to detect AMH in saliva, and like HHV-6, it's not detectable in saliva. I mean, really, really ridiculously high quantities of somebody who's has like extreme cases of PCOS were detectable, but not in the routine population. But it is detectable in hair, so we're working on a patent for that.
Oh, that's really interesting.
Yeah, the hair testing is used in so many different things. So yeah, we thought that was kind of a very easy way to get patients, because we can also run all the other steroid hormones off of the hair sample. So we can do a pretty decent profile, and we need more research too. I mean, our N's a little bit bigger than some of the HHV-6 stuff because we do a lot of our research in Spain. So it's just very low cost to get some of that R&D done with our Spanish partners. The cost you have to pay the patient is a lot lower than what it is in the US.
To get,
yeah. So like, instead of $100 gift card, it's only $25. Yeah. And there's in case, and this is an opportunity for all of us too, in the work that we're collaborating on together. Patients in Spain have a culture of being donors kind of for everything. It's very easy to recruit egg donors. It's very easy to recruit organ donors compared to US. It's easy to get people to participate in research. There's just a cultural, of course, I'm going to do right by my fellow human there, as opposed to here in the US, and so Yeah, it is very interesting. We've always been really good at recruitment here, but it is even easier in Spain. So if we get this partnership going with John Ace-Peruia, you know, and get him distributing with your lab, we could probably get some faster data even. Yeah. So lots of opportunity for our collaboration for sure. Any other questions? Kirby, Jody, Jessica, Jill?
Well, I mean, my interest is really more in what would be the benefit and whatnot as in the unexplained fertility, infertility patients, because that's kind of, you know, what our practice focus is. And I'd really like to see more data in that regard, you know, because what our goal is to reduce the amount of patients who go through IVF.
Absolutely.
To try, so if we have such a large load of the virus in unexplained infertility patients, I mean, that's where, I mean, I'd like to see our focus be driven there. You know, as far as protocols go, I mean, you know, I mean, goodness, even to the point, if we have somebody with unexplained infertility, do we just go ahead and pre-treat them empirically without even a test and see what numbers look like after that? So that would be, you know, I mean, that would be my vision as far as our practice goes. You know, what can we do to figure that piece of the puzzle out? You know, because right now we're trying to figure the piece of the puzzle of trying to, and I think HHV-6 would fall under this, but our focus is really trying to identify and treat chronic medical conditions and improve fertility naturally just by addressing those issues, improve pregnancy outcomes by addressing those issues. So a chronic herpes infection in the uterine lining would fit into a chronic disease. So,
Exactly.
you know, so I think, you know, the IVF data is fascinating and, you know, in that regard, but I would like to see the unexplained infertility people.
Well, yeah, I agree. I think that is the limitation right now is that we have the IVF data. I mean, it's because the providers, because they know the condition, they know the embryo is of good quality, they know all the stuff about the patient, they know exactly when the implantation is and stuff. So the data is really clean with if we want, and I know an REI who, because he didn't have an IVF practice, that's exactly what he did. He just started treating patients, and he felt that he was having better outcomes. I think the problem with that is that one, you're probably treating a lot of patients who don't need to be treated, because if you look at our data, even with the I think it was the ACOG study where when you get away from the recurrent implantation failure and you look at kind of a broader general, you're seeing like 17 to 28%. So that would mean you're treating 75, 80% of people who maybe would not benefit from it. So, and that's not preferred, even though acyclovir is a fine drug. And then I think it's just because there is a way to test for it and a way to quantitate. And the other thing is a way to identify whether it's HHV-6A or B. I've not ruled out B. No, people just haven't been doing the typing. I think it's most likely A, but we haven't done the typing. And the issue with HHV-6B is its sensitivity to acyclovir is about 3-fold less. So in those patients, you may want to consider, maybe you would have them on a higher dose of acyclovir for at least a period of time. So I think there's value in testing and getting more specific on an individual basis. But for all of us, the unexplained infertility are the ones you want to start there. We don't want to start with the IVF. I mean, that's not where you prefer to begin examination for the virus, because perhaps and hopefully many patients, if the virus is just enough to keep them, to reduce their fertility to 20% versus, you know, or 3% for cycle versus the normal, whatever it is, 30%. I mean, those are the patients that we would try to reach, because I think a little bit of help might go a long way in them. It's a low-hanging fruit.
We have an opportunity to get a grant from Torrance Memorial in Southern California. Thanks, Jessica. And so I'm wondering if maybe this would be a good idea to present this as the grant option. They didn't want us to do medical nutrition therapy because they already have a really strong medical nutrition therapy practice at Torrance Memorial. But maybe we approach it and say, you know, look, we already have pre-existing data on that points to, you know, this virus and its association to unexplained infertility. Let us be the ones that, you know, consult the patients, get the testing done, get the reporting done, and treat them to kind of prove that out, so it becomes even standard of care for Torrance Memorial. To get some data. What would be our goal, Kirby? Like, you know, what's the end for us to get comfortable with even, you know, like publicly marketing, like that we have a treatment for unexplained infertility? Because it's a big claim without the data. So I'd be hesitant to start making a public marketing claim. Like I want it to be part of the practice, but there's going to be some hesitation on like publicly making claims that we can, you know, solve for unexplained infertility. But
you need data. You need outcomes.
But what would be our goal of like what's the starting end? You know, do we need 100? Do we need 1,000?
Well, I mean, you need at least 100 to 200 patients. Because I mean, and unexplained, so all their testing's normal.
Right.
Yep. So, I mean, you could gather those patients fairly readily. But I mean, your outcome, I mean, you have whether it be two initial outcomes. I mean, number one, you want a clinically confirmed pregnancy, but then the second thing you want is a live birth. You know, that's your two primary outcomes. But you don't want to make a claim without that data.
Connie, along the lines of unexplained infertility, can you repeat what you said with one of your earlier slides about whether there's a relationship between HHV-6 and endometriosis? And I'll tell you why I want to know that in a second.
Yeah, where was that? Was okay. I'm just kind of looking. That was this one. Are we still on screen share?
Yes.
Okay, that was the endometriosis was done with Mike Doody. Oops, sorry. Where did you go? Honestly. Okay. And because I had a theory because I know from our transplant studies that the virus interferes with integrin activation. So I was thinking, oh, the cells are going elsewhere because they're mixed up with where they need to go, right? Where they're homing for, and they're going to peritoneum and all places where they shouldn't. With the paired data, and so this is really nice because we do have the implant. It was 18%. With Mike's 3 of 7. We did another 8 and did not find another one, so it reduced the frequency. And so, the best I can come up with is, I think HHV-6 in those patients, it's somewhere between maybe 9 and 18%. The best data is this right here, because they were paired biopsies, right? There was the peritoneal lesion and then the endometrial. And in the 3 cases where we found 6, it was in both.
The reason I'm asking is that there are many people that believe that you cannot make a diagnosis of unexplained infertility or idiopathic unless you've ruled out endometriosis by laparoscopy.
Right. Right. But endometriosis is in and by itself not a death blow to pregnancy, right?
Right.
But it contributes. It's a contributing factor for reduction in infertility. Well, what if you had another straw on the camel's back? Would that also be something that could be enough? Having one thing is bad. I just suspect in many of these patients, there are more than one factor in what they have to deal with, even if we haven't found it. But
So what this shows too, though, I mean, now that we're finding endometrial lesions in the brain, even endometriosis lesions in the brain, then the virus is also now systemic and not in those 3 patients, right?
That's actually really I didn't think about that. That's kind of an interesting, that would not be good.
No, that would not be. I would assume that wouldn't be good. Yeah, and I'm thinking like for if it's an exacerbator, maybe is that the right word? But so maybe that or for those who have it really severe and have found it head to toe, but the fact that we're now learning that it can be everywhere is crazy, because that means that the virus is everywhere too. I wouldn't think that'd be good at all. I would want to know if I were those patients.
Yeah, I think one thing with the data from Mike Duty, what we found was the duration of the infertility was shorter. So they were a little bit different compared to the majority of the cohort. So maybe they were earlier, and maybe maybe earlier is, I don't know if it's better to be able to intervene earlier in the case of endometriosis, but certainly HHV-6 is probably not going to be helpful in this context. But it doesn't look to me like it's the driver. It's not the driver of endometriosis. And we were thinking, oh, perhaps the dysregulation with again cytokines, chemokines, and just adhesion molecules and stuff. Maybe the virus was disrupting and therefore facilitating, the errant epithelial cells to go elsewhere. And it does not look like that. It does not look like that in this small study. Its basic pathogenetic mechanism does not look to be HHV-6, but it's not that HHV-6 couldn't, again, have an additive effect in these patients with respect to, their difficulty in conceiving.